To medical concept. Today's topic is glycolysis. Everyone's know or we are already studied in 12th standard. Okay, now coming to full details about the glycolysis. First, glycolysis otherwise called EMP pathway. That means M10 Merhoff pathway. And this definition is glycolysis is breakdown of glucose to pyruvate. That means glucose is breakdown to form two substrate of pyruvate in aerobic condition. In this anaerobic condition is form lactate. The site of the glycolysis is it will happen in all tissue of the body, and this only one pathway is taking place in the all cell of the body. Other cycles are present in same particularly that organ and tissue, but this glycolysis only will happen in each and every tissue, and intercellular site is cytosol or cytoplasm, and the starting material is glucose and end product is two end product in aerobic condition, in anaerobic condition. In aerobic condition pyruvate, in anaerobic condition it is lactate. What are all conditions for anaerobic glycolysis? If we go to gym, do a vigorous muscle exercise, that's also will make a lactate formation. And hypoxic condition also in anaerobic glycolysis, and RBC and lens, they don't have the mitochondria, so it also happen in anaerobic glycolysis. Now coming to we see about the pathway. Glucose is a substrate of glycolysis, and pyruvate is the aerobic condition of product, and lactate is the anaerobic condition of product. Now coming to see about each and step by steps. Okay, now glucose is converted to glucose 6-phosphate in the presence of hexokinase. Hexokinase is present in all tissue, but if glycolysis is present in liver, the enzyme is glucokinase, and glucose 6-phosphate is converted to fructose 6-phosphate in the presence of isomerase enzyme. Isomerase is nothing but different structure but same function, and fructose 6-phosphate is converted to fructose 1,6-bisphosphate in the presence of phosphofructokinase, and the short form is PFK. In the fructose 1,6-bisphosphate, bisphosphate means the phosphate groups are attached in first position and sixth position, so it's called bisphosphate. If this phosphate is attached in same number of carbon, we can tell it the diphosphate. Okay, now coming to fructose 6-phosphate is give two product. One is glyceraldehyde 3-phosphate and another one is dihydroacetone phosphate in the presence of aldolase enzyme. This is a equilibrium step, and the glyceraldehyde 3-phosphate gives 1,3-bisphosphoglycerate in the presence of glyceraldehyde 3-phosphate dehydrogenase. This 1,3-bisphosphoglycerate gives 3-phosphoglycerate in the presence of phosphoglycerate kinase. Kinase is nothing but it gives a 1 phosphate, otherwise nothing, and this 3-phosphate glycerate form 2-phosphoglycerate in the presence of mutase enzyme, and 2-phosphoglycerate give phosphoenol pyruvate. Enolase is the enzyme to give a phosphoenol pyruvate. Enolase means removal of water molecule from compound. Okay, this phosphoenol pyruvate give pyruvate in the presence of pyruvate kinase enzyme. Okay, now I just give a one trick. Wherever the kinase is present, the kinase means give a one phosphate group. Okay, see, pyruvate kinase, phosphoglycerate kinase, and phosphofructokinase, hexokinase, and wherever dehydrogen is present, it give a one hydrogen. See, it give a one hydrogen for NAD plus formation. In anaerobic condition, pyruvate is converted to lactate in the presence of lactate dehydrogenase. This formation is, there is no formation of ATP. It is used from energy from glyceraldehyde 3-phosphate to form a 1,3-bisphosphate glycerate in the between the formation of NAD plus. It is utilized for the anaerobic condition. Okay, so there is no formation of ATP here. Okay, now we are see about inhibitors of glycolysis. 3 inhibitors of glycolysis: one iodoacetoate and arsenate and fluoride. Here you can see glyceraldehyde 3-phosphate dehydrogenase inhibited by iodoacetate and also arsenate. Enolase is inhibited by fluoride. Okay, this fluoride is used for glucose estimation, because if we test the glucose in blood level, it is present in certain minutes only. Glucose is then converted to pyruvate. So we just want to prevent the formation of pyruvate. So we add a fluoride here. Okay. Then we are see about energetics of glycolysis in aerobic condition and anaerobic condition. First we see about aerobic glycolysis. We know a kinase means it form a ATP. Okay, so kinase is utilized for one ATP and phosphofructokinase is also utilized one ATP in the formation of pyruvate. And glyceraldehyde 3-phosphate dehydrogenase gives one NADPH. Okay, and phosphoglycerate kinase also give one ATP and pyruvate kinase give one ATP. Basically, one 6-carbon glucose give 2 pyruvate. So we should calculate for 2 pyruvate formation. Okay, so we add a 2. In NADPH value is 2.5 ATP. Here is wrong, that is here 3 ATP gives, but it's wrong. Okay, so totally we have a not a 6 ATP, sorry, totally it give a 7 ATP. Okay, in aerobic glycolysis. At the same time we see about anaerobic glycolysis. In hexokinase it's give a 1 ATP for utilization, and phosphofructase is also give 1 ATP, and glyceraldehyde 3-phosphate is 0 ATP because it is utilized. See here, it is utilized for lactate formation, so there is no here ATP formation. and phosphoglycerate kinase form 2 ATP, pyruvate kinase form 2 ATP. Totally it have get 2 ATP. Okay. Now coming to see about the significance of glycolysis. Why we should study the glycolysis? What are all the uses for glycolysis? We know we have a lot of questions, but at the same time we have some answers also. Okay, I'll tell you what are all. Glucose is provision of energy. That means glucose is a mm, major use of energy in our body. So glucose is converted to pyruvate to form in the glycolysis. Okay, so it give energy in this pathway. In muscle, glycolysis provide more energy in oxygen condition. If we have insufficient oxygen, it leads to anaerobic condition, so it form a lactate formation. And we take erythrocyte and lens, there is no mitochondria, so that leads from major energy. Okay, and glycolysis is some give a precursor of the some compounds, for example dihydroacetone phosphate and glycerol D3 phosphate. These are all used for the another synthesis. Okay, and glycolysis is intermediate. That means 1,3 bisphosphoglycerate for production of 2,3 bisphosphoglycerate in rapport cycle in the RBC. It is used for oxyhemoglobin in tissue. This in one particular enzyme is used for this. This is very affinity for oxyhemoglobin. And we see about regulation of glycolysis. Here hexokinase and phosphofructokinase is the most important, but the regulator is pyruvate kinase is regulatory enzyme of glycolysis. Okay, here see glucokinase and phosphofructokinase, pyruvate kinase. It's are all induced by insulin. Okay, because if we take a food, it contains glucose. Glucose is induced by insulin. Okay, insulin is induced, and repressor is glucagon. Okay, wow. And already we see about the inhibitors. Okay, that's all today's topic. And we also discuss about the exokinase and glucokinase differentiation. Exokinases we already see present in all tissue, but glucokinase is only in liver. The action is the action exokinases act on glucose, fructose, mannose, but glucokinases only act on glucose only. Inducer is not inducible anything, but it's induced by insulin only. Okay, affinity of glucose is high, but here very low. KMP value is very low, but here high. Okay, and significance is when blood glucose level is low, glucose can be utilized by body cells. And glucokinase acts on when the blood glucose level is high. Okay, that's all today's topic. Anyway, I'll try to complete all topics here. If you want do one thing, please subscribe to my channel. I'll try to make more videos about medical concepts. Okay, finally summarize glycolysis. This is very important question in university exam, so we just want to study it. Okay, your presentation is very important. It increase your marks. Okay, we write it with subtitles. Okay, subtitles we should write it subtitles. Okay, synonyms, definitions, site, starting material, end material, and that pathway is we should write it clearly, and different we can use different kind of pens. Okay, and inhibitors and energetics we see about it. inhibitors and energetics and significance regulations. We will all see about it. Okay, thanks for your watching. Please subscribe to my channel. Until this is medical concept.